generation times in different cells vary. And we know this by watching cell division occurring in the laboratory. We know in cell culture, when the cells have been removed from the organism, and remember we call that in vitro, we know that when it comes to a fertilized egg, it takes about 24 hours for each round of cell division. That actually holds up. It's a good estimate for how long it will take most cells to divide. Again, in vitro, out of the body. This phrase actually translates as in glass. It's meant to suggest, again, that this is not done in the body, in the organism. It's done in the laboratory. The companion phrase is called in vivo, and that translates as in life or in the living organism. So, of course, we've done a lot of work growing human embryos in the laboratory. That's the basis of modern assisted reproduction techniques like IVF. But we're also capable of growing lots of other types of cells in the laboratory for research purposes, for medical purposes, and so on. And this work has allowed us to compare the relative length of each phase of the cell cycle in that approximate 24-hour period. So again, if we use the embryo as our example, if you break down the 24 hours in the laboratory that it takes for the embryo to go through one round of the cell cycle, this is how it breaks down. It takes between 6 to 8 hours for the S phase, and it takes between 30 and 45 minutes for the M phase. Remember, these two rarely change from cell type to cell type, so these are kind of set. Notice M is a pretty fast process, relatively speaking. When you look at the gap stages, G1 is approximately twice the length of G2 under ordinary circumstances in the laboratory. But remember, it's G1 that's going to vary from cell type to cell type to cell type. G1 can be very, very short in a rapidly dividing cell, very, very long in a slowly dividing cell. And in a cell that stops dividing, it will leave the cell cycle while it's in G1. It will go from G1 into G0 or G0. So in the laboratory, it's a little bit artificial, but it's given us some information about the relative lengths of the phases of the cell cycle. Remember, in the living organism, in vivo, the cell cycle may have one generation time under normal circumstances, but for many cells, it can be sped up, it can be slowed down, it can even be stopped. And that's because in the living organism, the cell cycle is regulated. Remember when we were talking about macromolecules, we said that proteins are very powerful things. And the cell spends an awful lot of time and resources and energy making sure that the production of protein is carefully regulated. Well, the same is true of the cell cycle. The process that produces daughter cells from a parent cell is a very powerful thing. And so the cell is going to spend a lot of time and a lot of resources and a lot of energy regulating the cell cycle. The events of the cell cycle must occur at proper times. Remember, we said when the cell cycle includes mitosis, that should only be occurring during times of growth or times of tissue renewal or times of tissue repair. It shouldn't be happening outside of those times. The events of the cell cycle also have to occur in the proper order. We need G1 to complete before S begins. We need S to complete before G2 begins, and so on and so on. Each phase has to be performed and completed before the next phase starts. And of course, conditions outside of the cell, in the rest of the organism, have to be optimized in order for the cell cycle to occur normally. This is a process, this cell cycle, that is tightly regulated. But like all biological processes, it's not a perfect regulatory system, and it can go awry. When regulatory mechanisms go awry, you end up with uncontrolled cell division. And uncontrolled cell division produces what we call cancer. We'll move on now to think about the ways that the cell cycle is regulated. It's important to know that we do not fully understand all of the mechanisms that are involved in the regulation of the cell cycle, but we do know quite a bit about what are called cell cycle checkpoints. So again, our understanding is likely incomplete, but we do know quite a bit about 3 specific times during the cell cycle that regulatory mechanisms occur. These are referred to as the cell cycle checkpoints, and they happen late in G1, they happen late in G2, and they happen during metaphase of M. Over on the right-hand side of this slide, you can see a diagram of the cell cycle. You can see G1 here, followed by S, followed by G2, followed by M, that shortest phase. Then there's cytokinesis. You can see that 2 daughter cells are produced. Now, in addition to what we've seen before, we can also see these purple bands. There's one late in G1, there's one late in G2, and there's one in the middle of M. These are the checkpoints, and these are where regulatory processes are going to occur. What the checkpoints do is essentially control the progression of the cell cycle. At each checkpoint, a series of events will occur that will allow the cell cycle to be stopped if problems have been recognized. So during any one of these 3 checkpoints, the cell cycle can be stopped and one of 2 outcomes can be achieved. Either the problem can be repaired if it's not too extensive, or the cell can be sent into apoptosis. Remember, apoptosis is what we call programmed cell death, the natural set of steps that will result in the death of the cell. If a problem is detected at a cell cycle checkpoint, and that problem is too massive to be repaired, that cell will be sent to its death. And the reason that happens is because the organism cannot risk allowing an abnormal cell to divide. That could produce disease, significant disease for the organism. So we need to think about what gets checked during one of these checkpoints. Essentially what's happening, very generally speaking, is the cell needs to take stock that what should have been done so far has been done. Remember, we don't want these phases to occur out of order, and we don't want the next phase to occur before the previous phase has finished. So during G1, we're supposed to be replicating all of the contents of the cytoplasm, with the exception of the DNA. And at the late G1 checkpoint, the cell needs to make sure that that has happened. We also need to make sure at the checkpoints that the cell is prepared to go into the next phase. If we're at the end of G1 and we've made sure that everything that should have happened has happened, the other thing the cell has to know is, are we ready for S? Do we have the materials ready to perform S correctly? One of the most important things that the cell has to take stock of during these checkpoints is the DNA. Remember, the DNA molecule, out of all of the other molecules in a cell, is going to get passed on to the next generation of cells and is going to have an enormous impact on the health and functioning of those cells. So the cell has to make sure, number one, that it has been fully copied. Number two, that it has been correctly copied. And number three, that it was undamaged during the copying process. Now the next question is, how do these checks happen? We know, generally speaking, what has to be checked. How does the cell accomplish this? How does a cell know whether things have happened? How does a cell allow for the next stage to begin? This is a process that involves first messenger signals communicating with the cell. So things like hormones, growth factors, etc. Then we need those first messengers to activate second messengers inside the cell. And those second messengers are often involved with these regulatory proteins. So we need signal transduction to occur. The first chemicals that interact, say, with receptors in the cell membrane have to activate second messengers that will allow for regulatory proteins to be activated. It's often the regulatory proteins that are allowing for the next phase to begin. So when problems are detected, the best way to control the cell cycle is to prevent the activation of these regulatory proteins. And when that occurs, the cell will exit the cell cycle and go into G0. Repairs will either be made and the cell cycle will continue, or the cell will be sent to its death. Now, there are many individual pathways that have been described in terms of cell cycle regulation. And in terms of these three checkpoints, we're not going to have an opportunity to talk about all of them, but we will talk about a couple of the ones that have been well described. For example, during the late G1 checkpoint, there is one of those regulatory proteins called the G1 CDK cyclin. This is only one of several cyclins that show up at each of these three checkpoints, but this is the one that we'll talk about in some detail. When G1 CDK cyclin is active, it will regulate entry into the next stage, which is the S phase. Now, CDK is an abbreviation. It stands for cyclin-dependent kinase. This is a kinase enzyme. You know what kinase enzymes do. Kinase enzymes phosphorylate proteins. So, CDK must be bound to another protein called cyclin in order to be active and functional. The G1 CDK cyclin is going to go on to phosphorylate several different proteins, including one called RB protein, capital R, lowercase b. And this is going to start one of those cascades that we've seen before in cell biology. What RB protein is going to do when it gets phosphorylated is it's going to release a transcription factor. It's going to let go of a transcription factor called E2F. Remember, all a transcription factor is a molecule that is capable of binding to the DNA and altering gene expression somehow. When this E2F transcription factor is released, it will interact with the DNA and allow for the transcription of genes that will produce proteins needed for the S phase. G1 CDK cyclin is a regulatory protein, and like most of the regulatory proteins, its role is to allow progression to the next phase. On this slide, we're looking at a diagram of this G1 CDK cyclin process. On the left, you can see a blue linear structure that represents the DNA molecule. Inside this length of DNA are genes that contain the code for enzymes that are required for S phase to occur. Now notice that under normal conditions, there's a transcription factor called E2F that's bound to the DNA, and attached to that E2F is a protein called RB. Now, when RB is bound to E2F, transcription does not occur. In other words, something has to happen in order to allow transcription of these important genes to occur so that the cell can go into S phase. If that thing does not happen, at the end of G1, the cell cycle will just stop. Now, what kind of things happen? Well, they involve first messengers outside of the cell, things like growth factors, hormones, other types of signaling molecules. Now, this particular regulatory pathway involves what's called the RAS gene. Don't worry about that detail. Just know that it's the first messenger that's going to activate the G1 CDK cyclin. CDK must be bound to cyclin in order to be active. When these two things come together through this pathway, and this becomes an active regulatory molecule, what's going to happen is it's going to go out and phosphorylate proteins. CDK stands for cyclin-dependent kinase. This regulatory kinase is going to phosphorylate the RB. And when RB gets phosphorylated, it lets go of E2F. That's going to allow the E2F to change shape. It will interact with the DNA molecule differently, and that will allow gene transcription to occur. Those genes will be transcribed, they will be translated, and we will see that enzymes and other proteins that are necessary for us will be produced. And we will move from G1 into the S phase. If this doesn't happen, we will not move out of G1. It turns out that both the G1 and late G2 checkpoints involve these cyclin-dependent kinase, cyclin regulatory molecules. It's just that there are different types of cyclins and there are different types of CDKs. And each one phosphorylates a different type of protein and so has a different effect. What's interesting, though, is that the regulatory molecules at these checkpoints, the CDK cyclin regulatory molecules, are themselves regulated. if you think about it. They're regulated by the availability of this protein called cyclin. If there's not enough cyclin around, there won't be any CDK cyclin activated. And so we won't get the phosphorylation of protein. We won't get the push that we need into the next stage of the cell cycle. In addition to availability of cyclin, there's also additional phosphorylation that goes on of the CDK molecule itself. Other kinase molecules will phosphorylate CDK in order to make it ready to bind to cyclin. These are multi-layered, complex regulatory pathways. Remember, the cell is going to invest a lot of time, a lot of resources, and a lot of energy to make sure that cells do not divide unless everything is in place. The other pathway that we're going to talk about in detail is a pathway that occurs during the last checkpoint, which, if you recall, occurs during metaphase. Now, the purpose of the metaphase checkpoint, very specifically, is to make sure that all of the chromosomes are attached to the spindle fibers. Remember, during metaphase, chromosomes line up in the center of the cell. And metaphase is going to be followed by anaphase. And anaphase involves the spindle fibers shortening and literally pulling these chromosomes apart from one another. So it's critically important that each and every chromosome be attached to a spindle fiber. If there is even one chromosome that doesn't have an attachment to a spindle fiber, that chromosome will not be pulled to the correct location in the cell, and we will have an uneven distribution of chromosomes, which is obviously problematic for the daughter cells. This last checkpoint, I should say, during metaphase, will ensure that all of the daughter cells end up with the right number of chromosomes and therefore the right amount of DNA. On the bottom of this slide, you're looking at a cell right here that I'm outlining, and this cell is in metaphase. This is a beautiful image. I'm a little geeky, I know, but you can see the spindle apparatus right here and right here in dark magenta. You can see the chromosomes lined up in the center in purple. This is a centrosome. You can't see the centrioles in detail, but you can see that spindle fibers are growing out in all directions. There's another centrosome over here with a pair of centrioles in it. Spindle fibers are growing out. Here's the spindle apparatus. And at this point in the cell cycle, we need all of these chromosomes to be attached to these spindle fibers if the rest of this process of nuclear division is to go correctly. Now, the point of attachment between a spindle fiber and a chromosome occurs near this structure that's called a centromere. The centromere is the protein-based structure that connects two copies of one chromosome. Now, within the centromere, there's a specific location called a kinetochore. This is a term that you should be familiar with. The chromosomes attach to the spindle fibers at the kinetochore. So, at the metaphase checkpoint, any chromosome that is unattached at the kinetochore has to be recognized somehow. And the way the cell accomplishes that is that a lack of attachment is going to leave a group of proteins in place. In other words, there will be a failure to remove a group of proteins if the chromosome is not attached to a spindle fiber at the kinetochore. This protein complex includes two proteins that go by the abbreviations MAD and BUB. This complex of protein acts like a wait signal. These proteins are sitting there essentially to stall anaphase. If the chromosome is properly connected to a spindle fiber at the kinetochore, these proteins will be removed. This wait signal will be removed. Now, this is one of the mechanisms that is not fully understood at this time. So it's still considered a proposed mechanism, but most of the details are in place. This MAD-BUB protein complex is going to come together with a molecule called CDC20. And when CDC20 associates with this protein complex, MAD-BUB, the anaphase promoting complex will not get activated. So this is a checkpoint that involves a lot of not things. It involves not removing a group of proteins and not activating anaphase promoting complex. Let's take a look at this visually. Over on the left-hand side of this slide, you can see a drawing of a pair of replicated chromosomes. And they are joined together in the middle. Remember, this structure is called the centromere, and there's a region of the centromere called the kinetochore. In this sort of resting formation, there will be a complex of protein attached at that same location at the kinetochore. These are proteins including MAD and BUB. MAD-BUB complex, as it is associated with the kinetochore, is capable of binding to another protein called CDC20. And when the CDC20 is bound to MAD and BUB, it is unable to do its job. It's unable to bind to another molecule called anaphase promoting complex. This is the way everything is set up before the spindle apparatus attaches to the chromosome. So up until metaphase, before the spindle fibers are all attached, what'll happen is this yellow molecule will remain inactive. Now, once the spindle fiber attaches to this pair of chromosomes at the kinetochore, and that's what they're showing us right here, the MAD and BUB proteins will be released, and they will no longer be able to bind up and inactivate CDC20. So CDC20 will be released, it will bind to the yellow molecule, and it will activate it. So now this is active anaphase promoting complex. This active molecule is going to push the cell out of metaphase and into anaphase. As I said a moment ago, the two pathways that we've looked at in detail, the RB pathway and the anaphase promoting complex pathway, these are only two of what are likely many regulatory pathways that are involved in the regulation of the cell cycle. We know about the three checkpoints of the cell cycle, and we're learning more and more all the time about what specifically happens during those checkpoints to either allow a cell to keep going through the cell cycle or to stop the cell from continuing in the cell cycle. Remember, there are only two outcomes when a cell leaves the cell cycle. Either the problem is going to get repaired, or the cell is going to be sent to its own death. When everything is working well, a cell that is not going through the cell cycle correctly will be destroyed through apoptosis. Unfortunately, things go wrong in living cells. Things go wrong during the cell cycle, and these checkpoints sometimes fail, and they allow cells to continue through the cell cycle that should not be allowed to go through the cell cycle. One of the most dangerous things that can happen to a cell is to have its DNA altered, either mutated or damaged. That cell is a danger to the organism. Unfortunately, sometimes cells with mutated or otherwise damaged DNA escape these checkpoints, and those cells go on to divide when they shouldn't. Cells that are able to divide uncontrollably are the kinds of cells that create what we call cancer. Remember, there are also times when a cell is in the cell cycle that simple mechanical things go wrong. And as we discussed with the metaphase checkpoint, sometimes cells go through the cell cycle and divide and produce daughters that have the wrong number of chromosomes. The DNA can be perfectly normal, but the daughter cell inherits the wrong number of chromosomes. Now, this is not going to cause cancer necessarily, but this is going to cause disease. Uncontrolled cell division and incorrect division of chromosomes are two problems that cells work very, very hard to prevent. And that is why cells invest so much time, so many resources, and so much energy trying to prevent them.